Hi Surinder, we'll start in a few minutes once others join. Thanks for joining.
Okay, okay.
Hi Ignatius, we'll start in a few minutes once others join. Okay, we'll start now. I hope my screen is visible and I'm audible.
Yeah. Yeah.
Yeah, yeah. So this week we'll be discussing the last set of problems for week 8's assignment. So, 1 minute, let me turn on my camera. Yeah, yeah. So, for in this week's it was majorly case studies as to how to produce compounds of interest. So the first question is matching between chemicals and the cell factor, which is the kind of cell or the strain which was specifically used for production. The chemicals listed are lysine, 1,3-propanediol, 7-ADCA, and isobutanol. And the cell factories are E. coli, Penicillium trisogenum, Corynebacterium glutamicum, and Saccharomyces cerevisiae. We can take a couple of minutes and then try to come up with the answer for this.
So,
Yeah,
yes.
yes, Surendra, you can go ahead.
So, advanced biofuels are produced in the Scaramucci cerevisiae, like
Okay,
isobutanol,
okay. What do you mean by
and
advanced biofuels? Like, would you want to like brief on that?
uh, there's some disadvantage in the ethanol, na? Using of ethanol as a biofuel. So, to overcome the disadvantage of the ethanol,
Okay,
isobutanol and butanol they are used. So they are called the advanced biofuels.
hmm, like second generation that way. Okay,
Yeah, 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 yeah.
right. So,
Mm.
hmm,
And lysine is produced in the corynebacterium.
glutamicum. Okay,
So For me, A is the correct answer. Na? 1,3 propanediol, it is produced in the Escherichia coli.
Okay.
And 7 ADCA. So what is this ADCA?
So that is a kind of cephalosporin. So it can be, it's a precursor for cephalosporin. Cephalosporin is a particular class of antibiotics.
Mhm.
So these are naturally produced in a few organisms, like in this case, like Penicillium chrysogenum. Huh. So ADCA is one particular class which has specific modifications at positions. So this is being produced by Penicillium chrysogenum. So three is B. You said you go with option A. Okay.
Mhm.
Huh. Let's ask Ignatius as well. Yeah. Ignatius, what do you think about this?
Go with it.
Sorry, can you repeat? That was not clear.
Yeah, even I would go with it.
Even you would go there. Do you want to add something that either I or Surinder had missed out?
It's just that I know that isobutanol is actually uh, like it is taken from Helicocystis. That is one organism that I use.
Isobutanol or isopropanol are you talking about? Which one? Which one is it?
Butanol, like
Isobutanol.
isobutylaldehyde, also extracted from Helicocystis, the cyanobacteria.
Okay.
So
Hmm.
out of the given options here, I'm not really sure, but I think I will go with the.
Okay. Hmm. Right. Yeah, like it was discussed, the right answer here also is shown as A, where lysine is produced by Corynebacterium glutamicum. 1,3-propanediol is being produced by E. coli. Then 7-ADCA is being produced by Penicillium chrysogenum. And isobutanol is being produced by Saccharomyces cerevisiae. Uh, yeah, the next question is Sanofi Aventis has successfully produced Dash, an anti-malarial drug, through metabolic engineering. And the options are quinine, uh, artemisinic acid, pyrimethamine, and 4-amino quinoline. Can take a few minutes and then try to answer this question.
So during the means throughout this course,
Yeah,
I heard artemisinic acid produced through metabolic engineering,
huh.
but I don't know it is produced by Sanofi Ventis or not.
Uh, that is
Company name
okay.
I don't know.
Yeah, that
Company name I don't know.
right. So what we need to focus
It
on
was
here is,
actually
huh, go ahead,
so I
Ignatius.
think it was yeah, I think it was actually developed by Amaris, and then I think Sanofi again just took the same
Acquired it.
technology and they
Yeah.
yeah.
Hmm. As far as I remember reading from papers, it was, I think, J. Kiesling's lab, which had pioneered metabolic engineering in yeast, where they were able to bring in all the related genes and were able to express these genes required to produce artemisinic acid, which is an anti-malarial drug, in Saccharomyces cerevisiae. That is what I am aware
Yes.
of. Um, yeah, so what we need to focus on here is whether which of these is the anti-malarial drug and which of these was actually produced through metabolic engineering. And because this was the first successful attempt to produce a plant-based compound at a large scale, a very large scale in a eukaryotic organism like Saccharomyces cerevisiae, this still remains as a valid example. Hmm. We can leave out whether if it was produced by Sanofi or Sanofi Aventis or
So
Amyris.
it will be hard.
Sorry.
Uh, it will be hard to remember for the final exam.
Yeah,
This type of question.
these are all like memory based questions.
Memory
I
based,
I
yeah.
I really don't think you will have a major weightage for memory based questions, but we'll have a major weightage for the kind of problems that we used to solve with respect to stoichiometric matrix or reactions, gene knock-ins, knockouts, how to
Mm-hmm.
bring the isotope distribution vector, more of that sort. What where you will be able to apply what was taught rather than remember everything that was taught. So as long as
Mm.
you're able to understand why a particular method may or may not apply and how to apply a particular method for a given problem, I think we are good to go. And
Mm.
yeah, so did any of you receive an email regarding a summary session that we'll have tomorrow evening by any chance?
Mm.
No, sir.
Pardon?
Okay, so today will be the last problem solving session. Apart from this, we are
Okay.
supposed to have one summary session where I will be majorly how do I say, like summarizing all the course contents, and apart
Okay.
from which we can make use of that time to solve some doubts in case if you have any. So we thought we'll go through that phenotypic phase plane um, problem the other day. So apart from that, if we have any doubts, we can discuss it tomorrow. So I just wanted to know if any of you received an email regarding the same.
No, no, no.
No email was sent. Okay. Okay. As far as I know, it's tomorrow. But yeah, we can join in the same link at the same time, 6 to 8. That should work. If you have any specific doubts, we'll be just going through a summary of all the whatever was discussed.
Through this same link that we are doing today.
Yeah, yeah, the same link. Yeah,
Okay.
yeah.
Okay. Okay.
And so regarding last week's assignment, I think there was an error with one of the answers.
Okay,
So
you
will
had
that be
posted that in
rectified?
the Google Classroom, is it?
Right,
Mm.
right. Yes.
Discussion for.
Discussion forum. Yeah, sorry. I did remember seeing that. Give me a minute. Let me open that. So one minute. Maybe what we can do is once we'll finish this and then go through that assignment problem. So
Yeah. Yeah.
the thing is like I am involved in discussing the how do I say a sample set of problems, but I'm not directly involved in setting the assignments. So if we post it in the Google group, that's how we'll get to rectify the errors. But we can discuss that and try to see if what is the possible right answer or wrong answer. That should be fine, right, Ignatius?
Uh, yes, sir, yes, sir.
Excuse me, sorry. Yeah,
And also one more thing,
huh, yeah,
with regard to the production of this artemisinic acid,
yeah,
you said that they use S cerevisiae, right?
yeah. As far as I remember, it was in Saccharomyces
Yeah,
cerevisiae.
but in this page, there was this question. What is the probable reason they are using this S cerevisiae? So
Okay,
there are reasons such as ease of getting
sorry, go ahead. Your voice is breaking. If you can move to a better place, it will be helpful. But yeah, you continue, and then I'll try to explain why specifically Saccharomyces cerevisiae.
health.
Yeah,
Yeah,
yeah. Please continue.
yeah. So that's what I was a little confused with that. because around two or three options both seemed appropriate. So like which could be the particular option for that question?
Uh, so you're asking for the reason why Saccharomyces cerevisiae is used as a choice? Is that the question or
Yes, yes, yes,
okay?
yes. That is the question. But there are two or three appropriate choices that we could go for. So like which one would be the most appropriate? That's my question.
Okay. So one thing that I am aware of is that all of these are naturally produced in plants or higher order species. Right.
Right, right.
So, uh, for example, this artemisinic acid, it is produced from this organism called uh, Ar- Artemisia annua, and this Artemisia annua is it's a plant. It's an eukaryote. So one basic thumb rule in metabolic engineering is that when you have genes from eukaryotes, which give rise to proteins that are not very well characterized, you don't really know what are the kind of post-translational modifications that are essential for the function of that protein in that eukaryotic environment. So the minimum that you can do to ensure that the protein remains functional is to express this particular gene to form a protein in an eukaryotic environment so that at least a protein is functional. That is a specific thumb rule with respect to metabolic engineering. The That is one appropriate answer that I can think of. If there are other appropriate answers, you can tell me, we can discuss it further.
No, but that I would agree with you, sir. But that was not an option actually with regards to the modifications. There were options like higher artemisinic acid content, or then there was tolerance to fermentative inhibitors, and then ease of genetic modification. Such options were there. So, like out
Okay.
of those,
Was this a part of I mean, is this a part of this week's assignment or how is it?
right? Yes, yes, yes.
This week's
It is
assignment.
a part of the feature.
Okay. We can't really let out the answer. But we can maybe go through it once we are done with it. Another thumb rule
Okay.
in with respect to metabolic engineering is, like we had earlier discussed, maximizing the precursor content within a cell. So,
Mhm.
uh, if what I remember is right, there is something called farnesyl pyrophosphate, and in Saccharomyces cerevisiae, this was actually overexpressed for production of artemisinic acid. So I could not clearly listen to the one of the options that you had mentioned where they were talking about precursors, but I don't know if they had actually meant that as well. And yeah, so what we will do is first we'll finish these sample problems and maybe have a look at the last week's question that you had posed, and then come to this if that's okay with you.
Yes, yes, sir.
Yeah, yeah, yeah. So the answer here is artemisinic acid. Third question. State if the sentence is true or false. In biofuel production, the volume of the product plays an important role, while in case of commodities and natural products, the quality of compounds are of prime interest. So, yeah, we'll take a couple of minutes and then decide which is the right answer.
So
Yeah.
in this case,
Hmm.
obviously for biofuel production, volume of the product matters. We need
True.
more biofuel.
True.
So okay, and while in case of the commodity and natural products,
Hmm.
quality of the compounds are of prime interest.
Hmm.
So yeah, quality also matters.
So would you
And
say
here
it's
compound means
yeah.
these commodity and natural products or biofuel. I'm,
Here
he's asking
compounds
for
means commodities and natural products. Yeah.
quality of the commodity and natural products.
Yeah.
But it is of not prime interest because we are developing the E. coli that are resistant to the inhibitors like lignocellulosic crops. Okay, they are having many inhibitors. Then don't allow the growth of the E. coli. But we are developing the E. colis that are resistant to these inhibitors like vanillin is there, one of that compound vanillin. Okay, so quality may not matter because if we are going to improve the quality of natural products, cost of the product will be higher. Cost of the biofuel will be higher. We
No,
think
no.
of
So
that.
what they are saying is these are two different cases. One is biofuel production, where they say that volume is important, whereas in other commodities
Yeah.
and natural products, the quality is more important than the volume or the quantity. So in one case it's quantity, one case it's quality. That's what they're trying to say, and they're asking if this is true or false.
It is true.
Hmm. Um, Ignatius?
Yes, sir, it would be true.
Yeah. So this was, I think, discussed in lecture 33, where there was a discussion or this where the professor had called it as high value, low volume compounds and low value, high volume compounds. So biofuel, the aim of production of biofuel itself is to meet the demands of fuel consumption. So where even if there is a reduction in the quality or some adulteration or impurity, still as long as we are able to produce maximum volume, then that should serve the purpose. Whereas when it came to commodities and natural products, they had examples of uh, products like curcumin, flavonoids, which can be consumed by us and which have some potential medical value. In these cases, the quality of the compounds were of prime interest rather than the quantity. So this statement is true. Yeah. Huh.
But sir, are they always inversely related?
There's no inverse relation. What is being said is okay, you can think of it that way. Whenever you try to purify a compound to the maximum extent that you could, which is like 99% purity or beyond that, because of the kind of methods that you use, you will have a lot of losses during the process itself. So as you increase the number of purification steps, the final yield will reduce. As the yield is reduced, and because there is high demand, it is of higher cost. But as the yield is reduced, the quality is also improved. So you can think of this
Yes.
as an inverse correlation. True.
Okay.
Yeah. The next question. Identify the main steps involved in the conversion of lignocellulosic biomass to ethanol. And here we have options like feedstock harvesting and logistics, pre-treatment, enzyme saponification, fermentation, and production. So we can take a couple of minutes, and before we start with labeling which answer is right, first we'll discuss what each of the steps in a particular process corresponds to. Then we'll see which of the answers is right and why a particular step is needed. Yeah, we'll take a couple of minutes.
So,
Hmm.
feedstock harvest means cutting of the crops
Yeah.
from the field.
Yeah.
Then pre-treatments means we have to remove the unwanted compounds.
Hmm.
For example, if we don't require the lignin in the lignocellulose crops, so we have to remove the lignin. It
Yeah.
is called the pre-treatment.
Right.
And enzymatic treatments, we have to add the enzymes to degrade the cellulose, to degrade the hemicellulose into the hexose and pentose sugars. And thereafter, we do the fermentation with the help of the microorganisms.
Hmm.
So, so it is the way to produce the ethanol.
Right. Right. That was specific. Yeah. So As you had mentioned in feedstock harvesting, you just collect the plants, gather them together, and during pre-treatment, because we want to remove the lignin content from the lignocellulosic biomass, treat it with we remove lignin. We have both physical methods and the chemical-based methods. Then we have enzyme saponification, where we break down the entire cellulose into simple sugars, their corresponding monomers. And during fermentation, these simple sugars are taken as precursors, and the cells are also genetically modified such that they can overexpress genes required to produce ethanol. So, yeah, the right answer is answer A, like how Surinder had mentioned. Yeah. The next question is energy carrier molecules which are able to drive biofuel synthesis. And the options are ATP, NADPH, ATP and NADPH, GTP.
So here only NADH is required to produce the biofuel because from pyruvate,
Hmm.
uh, first step convert the pyruvate to the acetaldehyde by the help of enzyme pyruvate dehydrogenase complex.
Okay.
Pyruvate decarboxylase enzyme with the help of the
Decarboxylase converts
pyruvate
it to
decarboxylase
acetyl CoA, right?
acetaldehyde. Then
Okay.
acetaldehyde is converted to the ethanol by the help of alcohol dehydrogenase.
Pyruvate
And
is converted to acetyl CoA, isn't it? Acetyl CoA, it, when it's getting oxidized, it should produce acetyl CoA. Okay, you can go ahead. I'll think about it. Yeah, sorry. Huh. So you're saying pyruvate gets converted to acetaldehyde?
to the
Okay.
ethanol. And in this step, NADH is required. Only NADH is required. And in pyruvate decarboxylase enzyme,
Huh.
I think ATP is also required. I'm not sure about that.
Uh,
So I think
hmm.
both are required, and
Okay.
NADH as well.
Ignatius, do you want to try this?
Yes, sir. My answer would be both ATP and NADPH.
Okay.
Because one of key strategies used for driving biofuel synthesis, I mean, the production of like fatty acids that are relevant to biofuel synthesis is, you're supposed to like, down-regulate your TCA cycle and eliminate beta oxidation.
Hmm.
So the appropriate energy source required one would be ATP because of loss of oxidation of fat molecules. And
Okay.
apart from that, NADPH, it is like one of the main objectives, like cellular NADPH levels should be high in order to drive biofuel synthesis. So I would go with option C.
Hmm. Okay. Uh, yeah, I would agree with what you would say, specifically to talk about when, like how Surender had mentioned. Uh, for converting pyruvate to ethanol, like ethanol here is the biofuel that is, we can assume that it's the biofuel that's being discussed. In that process, two enzymes are required: pyruvate decarboxylase, which gives acetaldehyde, and alcohol dehydrogenase, which converts acetaldehyde to ethanol. So, in the second step here, NADH is converted to NAD plus. But to even get to pyruvate, we have other reactions which both produce and consume ATP. But so even I would go with option C in this particular case. Uh, huh. So yeah, in this way, both ATP and NADPH are uh, required for producing biofuel synthesis. The next question is PDC and ADHB genes that are successfully expressed in E. coli for high quantity ethanol production were obtained from. They have given four options: Saccharomyces cerevisiae, Zymomonas mobilis, Pseudomonas aeruginosa, and Brevibacterium KC. So, yeah, before we go to the right answer, first we'll try to discuss what these two genes are and where in the pathway do these specific genes play a role. Then we can discuss which organism it was taken from.
Actually, these are the same enzyme that we have discussed, pyruvate
Hmm.
decarboxylase and alcohol dehydrogenase.
Hmm.
Actually, in E. coli, na,
Yeah.
E. coli, these two enzymes are produced in very less amount, or they are having very high km value.
Hmm.
Okay, as compared to the other enzymes that convert the pyruvate to acetyl CoA.
Hmm.
Okay, that's why we have to transfer the genes in E. coli cells that are
Hmm.
having the more efficiency for the pyruvate, so that
Hmm.
pyruvate can be converted to the ethanol. Actually,
Right.
pyruvate is a branch point. Pyruvate is a branch point.
Hmm. Like you can
So
derive
it
many
can be
things out
converted,
of it. True.
it can go anywhere. It
True.
depends on the km value of the enzymes also. Okay. So the enzyme which is
Km
having the
values of
value,
enzymes determine how specific how, the binding
how
affinity.
binding affinity.
Yeah, and how fast the reaction can proceed. Not just a binding
Yeah, yeah,
affinity. That like that would
yeah.
depend on Kd and Ka. Km would
Uh-huh.
determine how fast the reaction can take place.
Yeah.
Huh.
So in native E. coli cells, these
Hmm.
PDC and ADH, they are having higher km value.
Hmm. Okay.
Okay. Ethanol can be produced from the pyruvate to acetyl CoA, then acetyl CoA convert to the ethanol. But here the two NADH molecules are required.
Hmm.
Okay, that's why scientists have transferred the two genes PDC and ADH
Hmm.
from Zymomonas mobilis because they are having the lower Km value. So they
Hmm.
immediately bind the PDC immediately bind the pyruvate and convert it into the acetaldehyde. Then acetaldehyde is
Hmm.
converted to the ethanol.
Ethanol. Right. Um, Ignatius, do you have to add something that we missed?
I'm not sure if this would be relevant, sir, but I'll just give it a try still.
Right.
So, like E. coli is an enteric bacteria,
Huh.
so they can actually utilize all types of sugars, but the yield of ethanol that they produce is comparatively less.
Hmm.
While S. cerevisiae and Zymomonas both are ethanol so they produce high amounts of ethanol, but
Hmm.
they cannot utilize pentose sugars as efficiently.
True.
So that is why, since Zymomonas is a bacteria, the genes,
Hmm.
I feel the genes were transferred from this to E. coli in order to increase the yield as well as the utilization of sugars to produce better fuel yield.
Yeah, that point was actually relevant. It was also, I think, mentioned in one of the lectures. Good that you had mentioned it. as to two classes of bacteria, one that can produce ethanol producing ones, and the others that are enteric. And possibly that could be a reason as to why genes from these ethanol producing are being expressed in enteric organisms. That does make sense, yeah. And like Surinder had mentioned about the kinetics of the process, that also plays a role in deciding which enzyme from which organism has to be chosen. Yeah, so the answer is Xanthomonas mobilis. Hmm. Huh. The next question is, to enable xylose fermentation by yeast, which of the following strategies would be most appropriate? And the options are insertion of xylitol isomerase gene, improvement of xylose consumption. Option B, insertion of pentose sugar utilizing genes, improvement of xylose consumption. Option C, deletion of xylitol isomerase gene while overexpressing xylose isomerase gene. And option D, insertion of xylose isomerase gene, improvement of xylose consumption, and insertion of pentose sugar utilizing genes. So the first thing that we have to discuss while answering this question is, uh, if we are looking at specific uh, options here, why what that specific option corresponds to. Like for example, when we say there is an insertion of xylose isomerase gene, we need to like look at what xylose isomerase gene converts to. Like for example, it might convert xylose to xylulose, vice versa. So, and how that specifically would help in xylose fermentation by yeast. So, you can take a couple of minutes and try to come up with a rationale for how a specific uh, step in each of the answers as answer options is relevant to the question. Then we'll go ahead with the answer.
So xylose is a pentose sugar.
Right.
Okay, and this Caromaceae cerevisiae has less potential to use the xylose. Native cerevisiae have the less potential to use the xylose sugar.
Okay.
So, uh, so to utilize the to make the cerevisiae
Hmm.
cells to utilize the xylose, we have to add some enzymes. We have to do some modification, genetic modification in the cerevisiae,
Hmm.
like we have to insert the xylose isomerase enzyme from the E. coli. E. coli
Hmm.
having the xylose isomerase enzyme, but yeast is not having that enzyme, xylose
Hmm.
isomerase.
Hmm.
They are having the xylose reductase and xylose dehydrogenase enzymes. Two enzymes are there in cerevisiae.
Hmm.
So we can insert the xylose isomerase, and in single step, xylose can be converted to the xylulose, and then xylulose is converted to the xylulose 5 phosphate. by kinase enzymes, and then it enters the pentose phosphate pathway. So pentose phosphate pathway, it should be higher also, because we have to utilize the pentose sugar more. So pentose phosphate pathway should be higher, and obviously xylose transport should be higher inside the cell, so that cell take the xylose inside it and utilize it. Okay,
Hmm.
so we have to put the pentose sugar utilization genes. We have to insert the pentose transport genes also. Okay, we have to improve the xylose consumption also, and we have to add the xylose isomerase from the E. coli. So that is from my side.
Hmm. So you would go with the option saying that we need insertion of xylose isomerase gene, then have transporters to improve xylose consumption, and also insert pentose sugar utilizing genes.
Yeah, yeah, yeah.
Right. Yeah, Ignatius, would you want to add something to this?
Yes, sir. Okay, when we talk about yeast, there, like the pathway of them metabolizing xylose to cellulose is a little, uh, it's not straightforward. So there is a formation of an intermediate that is mostly toxic, like xylitol.
Hmm.
So we would want to reduce that. So in order to reduce that, we wouldn't add the xylose reductase or the xylitol dehydrogenase. So a better option and a faster option would be to insert the xylose isomerase, as in how bacteria metabolize xylose,
Hmm.
and yeah, uh, improve the transporter. I mean, modify the transporter proteins and insertion of pentose sugar utilizing genes. So option B.
Option D. Okay. Yeah, that would majorly summarize as to why the xylose isomerase gene has to be inserted and the requirement of uh, transporters, and also how this relates to the pentose phosphate pathway and the reason for requiring pentose sugar utilizing genes. Hmm. Yeah, so the answer is D. Huh. The next question is which of the following is or are used as common strategy or strategies for amino acid production through metabolic engineering? Option A, amplification of biosynthesis pathway enzymes for the target amino acids and release of feedback regulation of key enzymes by the target amino acid, reduction of byproducts formation and increased supply of reducing equivalents such as NADPH. Then option C is only B. Option D is both A and B. So what we need to consider with respect to amino acid production within an organism is that there are no different there are no individual pathways for production of the 20 different amino acids. Rather, these are branch points and branching from one major amino acid. Modification of one major amino acid give rise to another major amino acid. So, in that way, what are the essential modifications that are required? We have to think from that particular perspective. Yeah. We'll take a couple of minutes and then try to rationalize what the right answer is.
So for me,
Hmm.
to produce the amino acid,
Yeah.
for the production of the amino acid,
Hmm.
we have to amplify the biosynthetic pathways for that particular amino acid.
Hmm.
We have to remove the feedback inhibition if there is some available, if some acting on that particular pathway, we have to remove
Right.
feedback inhibition.
Right.
Also we have to remove the competitive pathway so that byproducts do not form.
Hmm.
And NADPH production is also enhanced because NADPH is used as a used in the biosynthetic reaction. Most of the biosynthetic reaction NADPH is required. So for me both A and B
Both
answer
A
correct.
and B. Right. Hmm. It is. Yeah. Yeah. Before me talking, Ignatius, do you want to add something to this?
No, sir. Same answer. Both A and B.
Hmm. Yeah. Yeah. Just like how Surinder had mentioned, to produce amino acids, we have to first ensure that the precursors are being produced, so that these target amino acids can be produced. And if these have any feedback regulation, that should be reduced. Also, other competing pathways that lead to the production of byproducts should be avoided. And in general, supplying reducing equivalents such as NADPH should be increased. So both options A and B, which is given as option D here, is the right answer. Huh. The next question is, to mitigate the problem of endogenous ethanol production in E. coli, best adopted strategies are option A, overexpression of pyruvate decarboxylase gene. Option B, PDC and ADHP genes are expressed in an open under constitutive promoter. Option C, overexpression of PDC and ADHP gene. Option D, both B and C. So what we need to take a note of take note of here is that whether overexpression of these genes is helpful or expression under a constitutive promoter is helpful. So when we say overexpression, we mean we uh, place it in front of a promoter that's under the control of regulating molecule called an inducer. Whereas a constitutive promoter would mean that that gene is active always. So which of these is better? The gene being active always or the gene being active when a particular molecule, an inducer, is being provided to the cells? That's the only thing that we have to think about with respect to this question. Yeah.
So, constitutive expression without any regulation. There is no regulation
So
over the gene.
when they say that these are expressed in operon under constitutive pet promoter, they mean that these are expressed as long as the cell is alive.
Yeah, yeah, yeah.
Yeah,
So, best strategy will be to express the PDC and alcohol dehydrogenase under the control of the constitutive promoter. It will be the best because there will be no feedback inhibition and ethanol will be produced in a higher amount because these promoters are not regulated neither negatively nor positively. They continuously express ethanol.
right. But that would feedback inhibition has only to do with the enzymes per se, isn't it?
Yeah, yeah, yeah. Feedback inhibition at the regulatory.
So here, how would you say that feedback inhibition can be prevented when it is being expressed in a constitutive promoter?
Feedback actually Opron will never be off if the product is more in amount.
That
Okay,
would vary
it
with
is not
operon
putting.
from operon. Sorry, can you repeat it? It was not clear.
There isn't just one way to study.
Yeah, Surrender. We can't hear you.
Yeah,
Yeah. Huh. So you were saying in an operon, as long as continuously the products are being produced, it will never be feedback inhibited. But that doesn't generalize to all operons. There are specific operons that have specific set of regulatory measures that would prevent the operon from being expressed, genes of the operon from being expressed. So, uh, so can you rephrase what you were mentioning? Because what you mentioned was not very clear to me.
so if we consider the example of the tryptophan biosynthesis operon.
Right.
So here the if tryptophan is not there, then cell will synthesize the tryptophan.
Yes.
If
True.
if tryptophan is produced in a higher amount, tryptophan operon will be shut off.
True. That is the regulation. Yeah.
That's the regulation.
True.
So in constitutive expression, there is no such kind of the regulation. So ethanol is produced continuously. That's why I'm choosing that option B.
Haha. Yeah.
Because if
So
ethanol is
that I
produced
get
in a
it.
higher, uh,
That I get it. I was just curious about the feedback mechanism that you were mentioning because feedback mechanism has only to do with the protein uh,
yeah, yeah, yeah,
directly and not at least in this case, when you have a different promoter, apart from a natural promoter, uh, they do, they do not really apply. And when you have these heterologous genes being expressed in a host,
Mm-hmm.
um, these feedback mechanisms apply only when you manipulate at the genome level itself. So, and that too at specific positions having regulation. So, yeah, so you are saying that it should be option B where these enzymes are constitutively expressed, meaning they're expressed all the time,
Because
uh,
yeah, yeah.
so that they're
We we cannot produce the ethanol very much high amount also, because it can damage the cells also. It can damage the
the membrane. True, true,
membrane
true,
of the cells also.
true. That is true.
So I choose the B.
B, yeah. What about Ignatius? Do you think overexpressing the genes would be helpful? Because these genes are not toxic to the cell, they don't hurt the cell. So maybe if they're overexpressed, as long as enough precursors are there, more amount of ethanol will be produced. So do you think it could be option C also, or only option C, or only option B? What do you think?
I feel both the options seem appropriate, sir, but more towards option B.
Okay. Why
Yeah,
would you
but
say
I
so?
feel
Like,
both, right, like the cells when they cultivated in the medium,
hmm,
it is under constitutive expression. So the carbon source, whatever they are utilizing, it is being continuously being transformed into the ethanol biofuel there.
hmm,
Yeah, so there's continuous, the fluxes are being, they're being transformed. So they start from the carbon source and they give out ethanol continuously.
hmm.
And the
So,
problem with the E. coli is native E. coli cannot, they can utilize sugars very well, but they cannot synthesize good amount of ethanol, the
ethanol.
byproduct, the end product. Yeah,
Yeah, like you had
so I
mentioned,
feel
these are not
B,
ethanolic organisms, rather these are enteric.
yeah.
Yeah,
So I feel B is more appropriate, but I think C is also a strategy that could be used.
right. We'll see what the right answer is.
C.
One
What is
minute.
the right answer?
I'll just check.
Okay, okay, okay.
Yeah, so the right answer is given as D, where both the constitutive expression or the overexpression in either case, as long as
Mm.
both genes are being expressed, endogenous ethanol production in E. coli is possible. So I think how we can understand this question specifically is that instead of talking about regulation, rather, what is the minimum set of genes that are needed to produce or to improve ethanol production in E. coli? Just having the PDC gene alone, will it improve? Or will having both the genes will improve? Maybe we can think of this question in that aspect. If we think of the question in that aspect, then both options B and C are applicable. Huh. The next question is state if true or false. Systematic and rational random approaches in metabolic engineering have the target genes to be engineered. So before we go on to the answer
So in
question,
systemic means
huh,
whole. Systemic
yeah,
means whole.
whole, okay.
So we have to consider the complete metabolism of the cell.
Hmm,
Okay, we cannot go for a single gene or single pathway like that.
right.
So
So,
here the
hmm,
target genes are not obvious in that case. Systemic means. So we are not going to target any particular gene here in case of the systemic approach.
so you will be targeting the entire cell and how a particular target will affect other off targets. Is that
Yeah,
what you
overall
would want to
improvement.
mention?
Yeah, overall cell improvement.
Overall cell improvement. And what about rational random approaches?
Rational means logical. and random because we are not targeting a particular gene,
Hmm.
so that's why we are saying it is the means random. Yeah.
Hmm. So would you say that the statement is true or false?
False, false, false.
False. And Ignatius?
Even I think it's false.
Yeah, do you want to brief on why you think this is false?
I'm not exactly sure, but I remember reading that when it comes down to metabolic engineering, one is a local metabolic engineering and the other one is a systemic method or something.
Right.
Where local, like they target entire, I mean, a single gene, a specific gene, while in the systemic pathway, they target an entire biochemical pathway and see what modifications can be done.
Hmm.
So there it's not known, where here it's known.
Hmm.
So when it comes down here, there is actually a confusion I've been having with this systemic and irrational random. Are they like considered as two separate methods or as a single method, systemic and systematic and random, rational random? I've had a confusion there. Is that like one single approach or is it two different kind of approaches that they mention it this way?
We can check that. As far as I know, like it was mentioned, this systematic approaches are where you consider specifics of the system itself, whereas rational random approaches are otherwise. So, uh, I think we can call this as two different methods itself. Maybe I can check that and let you know that tomorrow. I need to read on this schematic and rational random approaches. Right. Um, yeah, so with this we come to the end of this discussion of sample problems for week 8. What we'll further check is we'll first go through that Google, this discussion forum. Yeah, so here you say week 7's lectures, question 4. Okay, so there is a live announcement uh, that was let out today stating that there will be a class tomorrow from 6 to 8. So this will be the final session, joining the same link. We will go to the questions. Week 7, question 4. Strategies developed to control the chromosomal gene copy number include A. Chemical inducible chromosomal evolution. B. Recombinase assisted genome engineering. Option C. ARA plus PBAD. Option D. A and B. And option E. None of the above. So, uh, hey, Ignatius, you had a doubt with this question, right? You
Not a doubt, sir. The solution was prompting the wrong option is what I felt.
okay?
It
So,
was prompting that the answer was ARA and PBAG.
hmm,
Basically, they were used for transcriptional expression, right? Arabinose and PBAG was used for the expression of transcription.
right. When you want to
While
modulate transcription, you can use
yeah,
these two plasmids with different inductions. Uh, yeah, so that is not appropriate. Yeah, option C here could not be appropriate. Whereas using a recombinase might specifically uh, induce homologous recombination at a specific site, thereby restricting the number of copies of a particular gene in a chromosome. That is one way. The other is chemical inducible chromosomal evolution. I have uh, do not have a good idea about this particular option. If you can explain why do you think what is this option, chemical inducible chromosomal evolution, and uh, why or how it can specifically control the number of gene copies, then it will be helpful.
I'm not exactly sure on how option A works to like maintain the copy numbers, but I remember it from the lecture that like these were the list of strategies that were adopted to like keep the
Okay,
copy
so
numbers
this
under
is
check.
from the table in the first lecture, is it? Okay,
Uh,
where they had mentioned a list of possible
yes, yes, yes,
methods.
yes.
Right, right. I remember the recombinase assisted genome engineering method because it was straightforward. Understood how it would work. Um, okay, yeah. So that in that case, your option should be answer. The answer should be option D and not C, like you had mentioned.
Yeah, yeah.
Yeah. Huh. And yeah, we'll first. Apart from this, you wanted to check with week eight's assignment, right? We'll see
So
which,
before going on to that, could you just there's another question I had in week 7.
sorry, it was not clear.
Uh, there's another question I wanted to clarify in week
Yeah, yeah,
seven's assignment.
week seven. Yeah, let me know. Yeah, which question?
There was a question related to spacers.
Are you able to see my screen?
No, no, sir.
You're not able to see my screen? Wait.
No.
Oh, sorry. I thought it was being shared. Let me reshare it. Yeah, now is it visible?
Yes, sir. I had it out with question 7.
Seven. Spacers are comparable short variable sequences that are inserted in between the short DNA repeats of bacterial CRISPRs. Spacer molecules function to shield infected bacteria against recurrent viral infections. Regarding spacer molecules, which assertion is incorrect? So the options are short pieces of invading viruses DNA are digested and added to the CRISPR sequence as new spacers. Viral DNA modifies CRISPR sequence spacer regions by DNA integration. Option C, spacers act as a kind of genetic memory for earlier infections, and option D, none of the above. Yeah, so what is your doubt regarding this, Ignatius?
In a way, if you see, when a virus infects a bacterial cell,
Hmm.
there is some kind of integration of DNA that takes place. It's not controlled by the viral DNA, but there is integration of DNA to the CRISPR RNA. So I feel option D also wouldn't be incorrect in a way. In one aspect, if you try to view it, I don't think it is incorrect. So the answer should be none of the above is what I feel, sir. But yeah, they've given D as the option. So,
Okay, whenever a virus enters a cell, it can either undergo two of the following stages, either a lyse. lytic or a lysogenic phase. In a lytic phase, what happens is it makes many copies of itself, then the cell bursts open, and what happens is you have many copies of the new virus that is being produced. Here, no DNA integration happens, whereas in the lysogeny phase, that is where parts of DNA from the virus is being integrated into the genome of the host, thereby providing new genes with new functionalities to the host. That happens in the lysogeny phase. And this uh, is sequence specific. What is needed for product integration of the viral genome, genetic material, into the host genetic material depends on the sequence or the flanking regions in the viral genetic material. And although they have given that this viral DNA specifically modifies the CRISPR sequence spacer regions by DNA integration, uh, it may or may not be possible. We don't really know. But since they're asking specific to the spacer molecules, as to which one is definitely right and which one is definitely not right, maybe that's why they have given this answer as viral DNA modifies CRISPR sequence spacers by DNA integration. Uh, this is one way to think about it. The other way to think about this particular case is that, like I had previously mentioned about is the sequence specificity for DNA integration, the very fact that an, a microorganism is able to withhold memory of a past infection despite repeated attacks from various different viruses, in turn, it implies that um that region, which is called the CRISPR array or the CRISPR loci, is not susceptible for DNA integration by viral DNA. This is another way to think about it. If at some point of time the viral DNA is capable of integrating into the host DNA, specifically at the CRISPR site, that would alter the functionality of CRISPR itself, thereby leading to no genetic memory, and that organism would not survive. It wouldn't pass down. So the very fact that CRISPR has survived till now in organisms as a genetic memory itself stands as a statement to say that this, at least in this particular region, viral DNA cannot be integrated. This is one way of looking at it. But if you have any disagreements, we can discuss this further.
I mean, not with the recurrent infections, but at least the first time there is integration, right? So, like the first time it encounters a viral infection, there is integration of DNA into the CRISPR array.
So that totally
So,
depends on the yeah, Surinder. Yeah,
so I will interfere here.
yeah.
Sorry for that. Uh, Viral DNA modify means viral DNA means you are talking about the complete DNA.
That
We
is
are not
not clear from the question. They are saying viral DNA modifies the CRISPR region. So
viral.
they are not saying if it's a specific gene from the viral virus or the entire uh, genome of the virus. They are not specifying that.
So it is confusing, na? Because complete DNA they are not talking about the as per the option.
Hmm.
I consider it as a whole DNA, viral DNA integrated as such, like
Oh, so you're saying
prophase
the
in case of the
entire.
entire viral DNA is getting integrated. Otherwise, in option A, specifically mentioned na, short pieces of viral DNA.
That is that is where it is being digested by the organism, the host itself.
Yeah, but they have not mentioned the short pieces of viral
Yeah,
DNA.
here the length of the viral DNA is not mentioned. That is true. I
Yeah.
agree. Hmm. So what Ignatius is saying is that independent of whether it is being at the CRISPR site or not, it is being integrated. That's why I was telling Ignatius there are like two life cycles for viruses, the lytic and the lysogenic. And only in the lysogenic case where it is sustained within the cell for a much longer time, it gets integrated. Whereas in the lytic phase, where it just takes advantage of the host and produces many copies of itself. Um, so I still do not understand what your disagreement with it is. I do understand that viral DNA can be integrated, and but that integration requires a specific site. That is something that we all have to agree with, because DNA cannot be integrated into a genome from random sites. It needs to have the flanking regions, like it was discussed with homology recombination. So that is an essential part when it comes to integration of any gene into any genome. Uh, that means that within the entire organism itself, there are only specific sites where this could be integrated. And maybe the CRISPR array is not one of those sites. Maybe the sequence is what determining this. That's what I believe is happening. But we should discuss this. If you can uh, precisely say that again, why you still think it, you're not convinced, it would be helpful maybe to even Surendra and me, even he can try helping us answer.
Yes, so as you said regarding the lytic and the lysogenic cycle,
True.
that is not mentioned in the question here.
Sorry,
And if you do, that isn't the the kind of life cycle isn't mentioned in the question. Whether it is a lytic or lysogenic, it isn't being mentioned in the question.
but for integration you need lysogenic. That is the given.
Yeah, that is
Like
given.
in lytic cycle you have no integration. That is given.
But if we talk in terms of the CRISPR mechanism, does that occur in lytic or lysogenic?
So in CRISPR mechanism, I really don't think the bacteria allows the virus to take over. So it's neither lytic or lysogenic. The bacteria, so both lytic and lysogenic takes place only when the virus can take control over the host. And in that case, the bacteria would die. But when the CRISPR mechanism, where the DNA is digested, first the viral, although the virus DNA enters, it is not able to take control of the host. Its DNA is digested and then it's integrated. So none of these mechanisms, lytic or lysogenic, is uh, of discussion in this particular case. That is the first question that we have answered that. Yeah, you can go ahead further.
Yeah. So when the option says viral DNA modifies CRISPR sequence spacers a spacer region by DNA integration, I'm not talking about the kind of integration that would occur when a virus is there is exchange of genes through homology. I'm
Okay,
talking about the mechanism where CRISPR basically cuts your DNA, fragments it, and integrates it into the CRISPR array
right.
for memory. For memory. So I feel that is appropriate in the kind of way if you see it. So that wouldn't be an incorrect assertion, is what I feel.
Maybe here it's just to do with the language. Them stating that it is the viral DNA that modifies the CRISPR sequence, but rather it's not the viral DNA, it's rather the host machinery, CRISPR machinery. So maybe that is why they think that this assertion is incorrect. I understand what you're trying to say. At the end of this viral DNA is integrated into the CRISPR sequence by DNA integration. That is true. But the reason for the modification is not the viral DNA, but rather it's the CRISPR proteins. It could just be
Right, sir.
an issue with the language. Yeah, the way we interpret it. Huh. In that case, what you said is
Yes, sir,
right,
that's it.
except that it's not the viral DNA, but rather the CRISPR proteins. Huh. If do you have any other questions in this assignment? We'll anyways discuss this assignment as well.
Yeah, question number 8.
8. Which of the following is a potential challenge or limitation of the CRISPR-Cas system in genome editing? A. Lack of specificity. B. Inability to target specific DNA sequences. C. Difficulty in synthesizing guide RNA. D. Cas proteins being too small. Hmm. Okay, so they say lack of specificity. The only thing that comes to my mind when we talk about CRISPR-Cas systems and lack of specificity is the PAM sequence, because the PAM sequence is the one that helps in recognizing a particular loci within a genome, and the PAM motifs are usually NGG, if we are aware of it. Uh, uh, Surendra, are you there?
And protospacer, they are complementary to each other also.
Yeah, that is there. But if we just have the Cas9 with the tracer RNA, with the guide RNA, that alone isn't sufficient. The PAM is also needed. Uh, Even I don't think that lack of specificity is the right answer here. If you ask me personally, I don't think any of these are uh, appropriate.
If
All, huh,
if
sorry.
A is the if A is the correct, then B is also in the same sense. B option are also having the same sense. B option.
True. I would agree with you. I don't think lack of specificity is a challenge or limitation for the CRISPR-Cas system. As far as I know, it is specific. And like you mentioned, if this is correct, I mean, if this is incorrect, I mean, both actually mean the one and the same thing. So I don't think lack of specificity is the right answer here. I agree to that. Yeah.
So
So you can maybe
correct
put
answer.
there is
So it will be notified.
you can maybe put up,
So,
huh?
yeah. If we take a look at option B for this question, it says inability to target specific DNA sequences. But when we talk about the nucleus enzyme, basically, they do have they work based on having specific and if we're talking in terms of lack of specificity, I feel that is the most appropriate out of all the answers uh, because as you said, the PAM region that is there,
Has NGG
that is the
motif.
only that is it kind of only depends on that. So maybe
True.
that's what they're trying to say, lack of specificity.
True.
So
That's
if
what
that PAM
even
region
came up to
wasn't
my
there,
mind. That's what came to my mind. See, the presence of an NGG motif in an organism is very common. So, because you have two bases defined, only the third base varies. So, the probability of finding that would be much higher compared to the probability of finding three random bases. So, that's what came to my mind when they said lack of specificity. But I don't really know how this question was set, whether they were thinking of this particular NGG motive. But another thing that we need to take into consideration is that this NGG motive, it tells you the specific location. But apart from this, we have the CRISPR RNA, which is complementary to this region of our interest. So that is the one that makes the hydrogen bonds, and then the cleavage starts. So, although the spam motif can be present in many different sites, the CRISPR RNA bind the sequence that's complementary to the CRISPR RNA is not present in many sites, and it's usually present in one copy at max within a cell, depending on how the CRISPR RNA has been depending on the length of the CRISPR RNA. So I don't really know how they mentioned lack of specificity, whether NGG motif or the CRISPR RNA. But so I really don't know if this is the right answer. That is something that I can accept.
Actually, I have put this in the discussion forum also.
Okay,
I put the email to Neptel,
okay,
Neptel Sport.
okay,
So they asked me to put your queries in the discussion forum.
right,
So I have these queries, these four questions. I have the query week 2 and week 7.
okay. Hmm.
So if we are correct, then marks should be increased in the assignment section. Our marks should be either this question should not be considered, or if it is considered correct for all, it should be considered incorrect for all. So that
Right, I get it, but I am not actually involved in these markings or
okay,
framing
okay,
these questions. Uh, my involvement ends with this tutorial sessions. So
okay,
that's
okay,
why I had not specifically typed an answer here. I cannot do that. But
okay, okay,
whatever questions that you had, like this amphibolic pathway, active transport, thickness of the membrane, we had a discussion on all the questions that you had posted here. But I can't officially answer in uh, with only the course creators from IT can specifically answer to this. So that's why we had a discussion on the answers, but I had not typed a particular answer here because we don't have the authority to do that.
mhm.
So I don't really know if marks will be given to all the students attempting that question or that question itself will be removed from the paper. Uh, I think for one of these questions, there was an answer from the course creator itself for one of the questions that you had asked. Let me check. Yeah, for one of this question, there was an answer from the NPTEL team itself. So I think they would take some more time to answer. Not really sure.
Okay,
We,
okay.
yeah, you can maybe post this. Yeah, you have posted it, so I think it's up to them to answer. But yeah, we had the discussion, and that is the extent to which we can go. Hmm. Now we'll discuss the answers for assignment 7's questions. So the first question is identify the three basic steps of CRISPR-Cas system: targeting, synthesis of CRISPR RNA, and adaptation. Adaptation, CRISPR RNA synthesis and interference. Targeting, interference, and production of CRISPR RNA. New spacer is delighted from virus, integrated into CRISPR system. CRISPR RNA guides molecular machinery to target and specify viral genome. Production of CRISPR RNA. Uh, so here, adaptation itself would mean that the entered viral genomic material is chopped and then it's introduced within the cell. Then CRISPR RNA synthesis means based on what is being introduced in the repeat space or repeat motifs, this RNA is being synthesized. Then, when a new such viral genome enters, with the help of the Cas proteins, this CRISPR RNA binds to that viral genome or RNA, and then it cleaves. So the answer is option B. The second question is uh, you have different options like MAGE, CRISPR, zinc finger nucleases, tail repeat, and co-MAGE. And option B is which of the what is the particular advantage of these particular uh, methods. So here CRISPR is an adaptive immunity in bacteria 2 5. So option B is wrong. Then zinc finger nucleases are um, cuts the DNA within a sequence that separates two flanking zinc finger binding sites. Uh, okay, mages efficiency, less greater than 20 base pair modifications can be effectively introduced. Uh, co-mages, efficiency of homologous recombination for larger fragments are low. Yeah, so since not a majority of this was discussed, what I know from this is that CRISPR is the adaptive immunity and zinc finger nucleases cuts DNA. So 2 is 5, 3 is 4. 2 is 5, 2 is 5, 3 is 4, and this is the only option that is suitable according to that. I'm not sure about other methods. And the third question is editing cannot be done by using CRISPR-Cas technique at any site devoid of PAM. Okay. Yeah, so here they talk about the T loop, which is the loop that is present in the tracer RNA, and PAM is that NEG corresponding CRISPR array and specific protein complex. So, yeah, so what we can understand from this question is that when we have the CRISPR RNA, it can make hydrogen bond and interact with its complementary DNA, but that is it. For the cleavage to happen, PAM motif has to be present in the below the 3' region of the CRISPR RNA. So that's why maybe they've given that as PAM as the right answer. Then we discuss this fourth question. The fifth question: mutations in the NAC domain. Okay, REC is recombinase, is it? NAC is nuclease. Okay, may affect cleavage activity, both of which could impact the overall functionality of CRISPR-Cas system. Could affect the binding and cleavage activity, affect specificity, activates nuclease activity, does not affect the activity. So in one of the lectures they had mentioned what these different proteins are and how they have uh, different functions, and that is where they had discussed about this protein, the modification of this protein into dCas9, where it is a deactivated Cas9. There they talk about how nucleus domain specifically involves in uh, cleavage, and if you do those two mutations, uh, you will get rid of the nucleus activity. So the options that it does not affect the activity and it activates the nucleus activity is wrong. And this recognition motif is needed, affects the binding. So maybe that's why option A is given here as the right answer. Identify whether true or false. Even if mutations introduced into the nucleus domain, DCAS9 can still bind to the target DNA guided by a guide RNA. True. Like we just discussed previously, these mutations would prevent it from cleaving but doesn't affect its binding. Yeah, we discussed questions 7 and 8. Question 9. What does the acronym NGG represent in the context of CRISPR-Cas system? Uh, NGG is any nucleobase. G is guanosine. Another G is guanosine, and it's a part of the protospacer adjacent motif. So it's option D. Then, which of the following options best describes the significance of CRISPR arrays in bacterial genomes? Uh, they code for essential bacterial proteins. They provide resistance to antibiotics. They are involved in recognition of foreign DNA. They regulate cellular metabolism. Yeah. So here They don't code for bacterial proteins or provide resistance to antibiotic or against cell metabolism, but they recognize foreign DNA. So that's the right answer. Yeah, so with this, we come to the end of this week's uh, problem solving session. If you have any specific topics that need to be stressed upon tomorrow during the discussion session, please let me know so that I can have a higher weightage for those topics. Otherwise, I would give a summary of all the lectures that were discussed and we will be done with it. You can post if you have any specific topics to be discussed in the discussion forum by tonight, or even in the chat box here, you can post the specific topics that you want to be discussed. We can wait for 5 minutes till you finish posting, and then we can end the call here. Is there some specific topic that you want to be discussed tomorrow?
Oh, we'll cover every topic.
Okay.
Means every week assignment, na?
No, we'll
Overview.
we'll we'll not cover the assignments. We'll just talk about what all was discussed in
Yeah, yeah.
general, and we'll be that will be it. Yeah.
That will be better. In brief, we will discuss.
Okay, sure. So you don't want specific topics. Okay.
Yeah.
Okay.
So I just had a doubt.
Yeah.
Yeah, like for the final exam,
Ah.
for the proctored exam,
Yeah.
like will we have these problems on the basis vector and stuff? Because I mean, if MATLAB was it, then it would be easier. But
Right.
I don't think we would have access to MATLAB there. So like, how would we proceed with the calculation? Like,
So
do it
whatever,
manually?
yeah, whatever calculations that were taught to be manually practiced are might be expected. Uh, Nothing that would require you to involve MATLAB would be asked. I can assure you that. But whatever you was taught to be manually calculated, you can expect questions from those.
Okay, sir.
Yeah. Okay. Okay. Then we can meet tomorrow 6 to 8. Same.
And sir,
Yeah.
yeah, just one more thing.
Yeah. Yeah. Sure.
Yeah, for tomorrow's thing, there's one concept that I kind of had a confusion with,
Sure. Which one is it?
and that was with the reactions. You know, the coupling of reactions, fully coupled, and then uh, there are such reactions in the metabolic flux.
Uh, can you repeat it? Which set of lectures was it? Which week was it?
If I'm not wrong, it is in three.
Week
It should
three.
be in week three, week three or four. I'm not exactly sure, but yeah, it's in the first half.
So week three. Week three, I think regulatory networks and stoichiometric matrix, one D annotation, 2D annotation were discussed.
Yeah, I
Coupling,
think it should be after that then.
coupling. Let me open this. You can tell me which of these is it. In week three, we had till the reconstruction. In week four, we had flux variability analysis, flux coupling. This one is it?
Yes, sir. I think it's
Lecture
flux
18.
coupling. Yeah.
Hmm, one minute. Flux coupling. Okay. So you want flux coupling to be discussed. Okay.
Yeah, like they had given an example in the presentation, like in the lecture material, but I'm not
Okay.
exactly sure how they yeah, they arrived at that. Like which was fully coupled and which was not fully coupled and all that.
Okay. Sure. I will look at it and we'll have a discussion about it tomorrow. If
Yes.
there's something else that you want, you can let me know. You can let me know right away so that I'll prepare the slides accordingly. something that you would want to discuss, or if this flux coupling is fine, then okay. We'll have other topic discussions as well. Fine. We'll end the call here. Thank you.
Okay.
Yeah.
Thank you.
Thank you.
Thank you, sir.